we have been trying to correlate structure and function of skeletal, the musculoskeletal system in vivo in human beings for quite some time. And one of the main onuses of that is to do it non-invasively using a safe modality so that we can carry out longitudinal studies. And out here you see functional studies in various aspects using quite sophisticated MR techniques, as well as architectural or structural studies such as shown out here, fibrocartilaginous tissues inside the lower leg. This is subcutaneous fat. These are the extracellular matrix and fibrocartilaginous tissue. Out here we have entirely different application in the anal sphincter complex, in which you can see the functionality of the different very small structural units. And those units can again be elucidated very nicely by fiber tracking using a sophisticated MRI technique called diffusion tensor imaging. What we will be presenting today is our attempt at trying to elucidate the factors that affect the loss of force and contribute to the loss of force as human beings and other animals as well age. Out of these, the different factors like the neural drive has been well studied, as has been the loss in contractile muscle tissue with age, which is termed as sarcopenia, as opposed to the disproportionately greater loss of muscle force, which is termed as dynapenia. And both of these factors have been well studied. However, there are other factors which have not been as well studied. For example, what's the role of the extracellular matrix as persons age? This extracellular matrix is composed of both endo, peri, and epimysium units. And what we are postulating is the major loss of force occurs from lateral force of transmission of force getting reduced because of the remodeling of the extracellular matrix due to various different kinds of changes in the biochemical structure of the extracellular matrix. There are other factors like architectural gear ratio, which I'll not go into today. This loss of force from lateral transmission becoming less effective can account up to about 80% in animal studies by certain researchers. So the way we are, as I said, this remodeling of the extracellular matrix is a result of various biochemical reactions, again, which of course is the sum total of the talk of a lot of the other presentations out here. So I'll not go into any detail in that respect. What we'll focus instead are the pathways of force transmission, including the longitudinal transmission. That's what we are all used to. So a muscle fiber contracts, is attached to the tendon and causes the force to be generated at the end of the limb. However, there's the second mechanism, which is lateral transmission of force, in which the fiber contraction exerts a force on the adjacent endomysium, which in turn finally ends up in a lateral transmission of force. And because of the changes or remodeling of this biochemical structure, the efficacy with which that lateral force is transmitted reduces with age, and that's essentially what we'll talk about. We have developed various sophisticated techniques, including velocity encoded phase contrast MRI techniques, by virtue of which we monitor the strain, which you are all familiar with, the change in length divided by the total length, that would be strain. Strain rate, which is its same derivative with respect to time. How does it change with time? Why do we at all use strain rate? Because of certain computational ease, we don't require three-dimensional tracking, and baseline or reference state is not required. So we therefore compute both strain as well as strain rate in our work. The objectives of this, therefore, is to find out, to carry out these sets of experiment, both in young and senior cohorts, and look at what changes are manifest as the person becomes older and older. So for that purpose, in this particular presentation, we'll present results from 3D strain and strain rate tensor maps. Additionally, And very importantly, we have recently developed, and it's just come out as a accepted paper in Frontiers in Physiology, a novel accelerated compressed sensing VPC. And I don't want to dive into too complex a term. The whole point is that at one time, and all our previous papers, we would take about 2-3 minutes for each MVC that we get the patient to image on. And while that's easily tolerable by young patients for all these last couple of decades, for older people, of course, is going to be harder and harder unless you reduce the percentage of MVC that they have to exert to something like maybe 10-20%. But we want to go to higher percentages like 60. And in order to do that, we need to reduce that entire scanning time enormously or maybe less than a minute, which we have managed to with these novel techniques, as you can see out here, and we'll talk about that. And we will therefore study young and senior cohorts at different percentages of MVC, 30, 40, 60. And in this presentation, we'll present the results from isometric contraction. In others, we have also concentric and dorsiflex or eccentric contractions. We will present results only in the MG and the Solius, and we will present results in terms of principal eigenvalues, and I'll talk about what they actually mean, and invariants, which correlate with the percentage of MVC and to force. In other words, what are these complex things? Well, there's a whole host of computational parameters that are the end products. Out of that, we do a statistical analysis, and we'll present to you only those which correlate nicely with the percentage increase in maximum voluntary contraction and to the finally to the total amount of force that's exerted. So for this purpose, therefore, we had 19 subjects, out of them 11 young and 8 old. Most of them are 75, older than 75 years. Some actually, well, we don't present the results of those, 90 plus results as well. In other words, it's tolerated well because of the massive reduction of scan time. I'll not go into the technical details anywhere near the talk, but we use, as I said, velocity encoded MRI sequences, and we use that with isometric contraction at these 3 different levels of percentages MVC. We use 3 contiguous oblique slices, and we oblique them sagittally, and you'll see examples of that, so that through the MG, you can see some of the fascicles, because it's important to have the slice oriented just so that the fascicles are visible on that particular oblique sagittal view. These, the important ones out here is that it takes a total of about 12 minutes for 3 slices at 3 different, or that is 9 total dynamic study for both young and old. And therefore it's a very nice time and it was a good thing to develop this technique. Just one second, pointer. Okay. And we were able to do that by incorporating this very novel type of pulse sequences entirely done by ourselves, which uses a technique called compressed sensing. You would have heard all these buzzwords about machine learning and big data. This is a sort of offshoot, not quite, but it utilizes some of those technology as well. And one uses, again, I'm not going to go into techniques out here, but it takes about 30 minutes to post-process these images, all done on PCs and using MATLAB. All codes are written by us. We also had tagged on diffusion weighted images, which I will not present right now. Just to give you a little bit of a background, this is the fiber that we are talking about. During compression, these are, the EFF is what's the eigenvalue along the fiber axis. However, what we note over and over again is as the fiber contracts, you'd expect the strain to be along the fiber length. What we saw and other people have seen is that the change in length of the muscle is angulated to the direction of the fiber by a certain angle called the strain fiber angle, which is pretty strange, right? In other words, even though the muscle contracts, the actual strain or the change in length is at an angle slightly offset from that fiber direction. And this angle depends on age. And it is caused essentially by the extracellular matrix, hence the whole rationale of our studying this. And that's what we look in terms of tensor analysis. The E fiber out here is angulated to the fiber direction, but essentially connotes the amount of compression or elongation. And the E in plane that you see is in the cross section of the fiber, and therefore gives us an idea of how the cross section of the fiber changes as force is produced. This is the experimental setup in which we have the person lying inside the muscle scanner. At the foot of his foot, this is put in a cast, and the person is asked to exert force, continuous force, as given by these red target curves. And as he exerts or she exerts, the blue curves denotes the amount of force actually exerted. That's mathematically differentiated by our hardware to produce a trigger, so that we fool the scanner into believing that we are scanning the heart, taking a cardiac gated scan. Here's the patient lying inside the scanner room, a video projection of the target scan, which is this red scan. This maximum level is adjusted to 30, 40, and 60% MVC, as I showed before. This is the average of all these phase encoding lines as are used in MRI, the average of that with standard deviation shown out here. Within that, at the end product, we will have different types of dynamic scans. Here's the phase encoding one. This is the medial gastrocs. This is the soleus. The color indicates the extent of deformation of the muscle. And as you can see, it's maximum near the aponeurosis. We can use another technique in which we call it spin tag, which is often utilized for cardiac cinese. And as the person is exerting force or plantar flexing and dorsiflexing, you can see that you start with these lines straight, like so, and then those lines distort as the person is exerting force. And what you see out here is very nicely the mg differentiated from the soleus by virtue of the amount of and directions of the distortions. We can put seed points and look at how each point traverses. We can put these seed points next to the aponeurosis and tendon to look at compliance factor by monitoring how much those fibrocartilaginous tissues change. So I'm not going to go into the very complex method of acquisition and reconstruction that's shown in this flowchart. We utilize a factor of 4:3 variable density. We utilize coil sensitivity factors. It's a two-step joint reconstruction purpose, and we use a very complex method of analyzing the entire results. We put quantitative analysis for quantitative analysis. We put two regions of interest, one in the medial gastrocs and other in the soleus. And this is the pressure curve that I showed, out of which a certain portion is sampled. 17 temporal frames are incorporated into that to show the scenes that I just now showed to you. And within that, what you see out here are, in the center, you see the average force curve. These are the average force curves. These are at the three different MVCs as shown out here. This is the 60, 40, and 30. Point to be noted out here, not very visible, but, and I'll show it that into the next slide. So I'm going to toggle between these two slides. The MVC levels during voluntary contractions is significantly lower by 45% in the senior cohort compared to the young. Again, nothing very big. And that is if you were to compare these curves with these seniors, all right? Then the next one would be on the right-hand side, we have the corresponding strain color map. So the color out here indicates the amount of change in length at the peak value, only at these points, all right? Whereas on so these are the peak strain, sorry. And these are the peak strain lengths. These are taken at the middle of these, not middle, the maximum gradient values. And the point that we will try to convey out here is if you are able to, let's say, compare this curve with that in the senior, you'll see a lot greater amount of reds out here compared with the, sorry, in the senior, it's much less compared with the young. And that means the strain factors, that is these, in the fiber in plane and out plane, all these tensorial parameters that we are determining change enormous amount as you go from senior, from the young to the senior cohort.
5 minutes left.
Yes. Reduction in these parameters with age can be very easily seen. And the important significant carry home message is that the significant changes with age and percentage MVC, both factors are seen in the MG, whereas in the soleus, only age-related changes are seen. So this is a very important factor that we see that the medial gastrocs reflect changes both with age as well as with percentage MVC that's been exerted, whereas the soleus does not show any change related to percentage MVC, and that most of you muscle physiologists know, but it does show enormous amount of changes with age, all right? This graph I'm going to skip. This enormous table essentially just reinforces what I said. You have the young and the old, and we did a lot of statistical analysis. It's all there in the paper that I just now talked about. And let me go to the major conclusions. So we had hypothesized that the shear strain, that is the off-diagonal elements that, again, relatively a little bit of a complex, which is exerted through the endomysium, is the factor that causes this lateral transmission of force, and that changes with age. That's caused by this endomysium, the outside part of it being fixed to the outside matrix, but the inner part contracting along with the fiber, and therefore it distorts in shape, and that causes the shear strain that we are talking about, because the biochemical composition of this endomysium is changed with age. It becomes less and less effective transmission of force that's initially created by the contraction of the muscle compared with what's finally transmitted at the end of the limb. We actually, for the first time, were able to monitor in vivo in humans these different types of strain, negative strain being the actual compression of the muscle fiber. These two parameters, L2 and L3, if you cut across the fiber, that's what you see. And it essentially tells you and is able to tell you how much of asymmetry there is in the contraction or change in the shape of the fiber. That's very important, and this has been determined microscopically in vivo in the human beings. I will skip this one that's related essentially with what we are able to detect using this 3D volume tensor measurements. So our hypothesis was that shear strain measures the shear in the endomysium and can prove to be a surrogate marker of lateral transmission of force. This could be utilized in various pathological states, and that's what our future study will focus on. And we were able to achieve all of this pretty amazing amount of results, pardon me for using that adjective, simply because of this development of this novel technique, which allows senior people up to even 90 years old to exert and complete the study in less than 9 minutes. Each scan being about less than a minute with 1 minute rest in between. So our technique therefore will enable us to study people, particularly as they can change with exercise, various types of exercise. That's our next future work. And we can do a longitudinal study in senior people using this novel method of imaging.